I'm Dr. Simeon Boyd. My research is focused on non-syndromic craniosynostosis. Craniosynostosis is a relatively common defect, congenital anomaly, birth defect, that occurs in utero, but it's typically recognized in the first few months after birth and results in unusual shape of the head due to fusion of the sutures, zippers, between the flat bones of the skull. And that leads to increased intracranial pressure, cosmetic defects, vision difficulties, and some other associated symptoms. Because 1 out of 2,500 newborn babies have this condition, we have established a large collaborative team that I lead in order to find the causes of this condition. Our goal is to understand why it occurs and then to develop strategies to prevent it and potentially to treat it better, both surgically and long-term without surgeries by conservative methods. We have created a very large collaborative group. We call this International Craniosynostosis Consortium. Each of our collaborators contribute toward our common goal. At this point, we have recruited close to 1,300 families that have generously donated their time and information and biological samples. And with those samples, we were able to identify genes that can explain at this time perhaps about 10% of the cases. It's not very high percentage, but we're filling up this gap mostly with the resources provided by NIH and also by those provided by the Gabriella Kids First Pediatric Research Program. I'm the principal investigator together with Dr. Paul Romiti of the University of Iowa on a project that was awarded to perform whole genome sequencing of 400 families. This data is very voluminous. It really requires a lot of server space and expertise in order to process it. So at this point, really, we benefit tremendously from the expertise that the Data Resource Center provides to us.